2022, the Stockholm Resilience Center quantified the planetary sustainability limits for so-called new entities or man-made chemicals and found that they are already beyond the safe limits. And I quote their paper, "threaten the safe operating space for humanity." This is of course an important topic for Wetsus as we here work on developing sustainable solutions to protect human health and to protect our environment via topics such as PFAS removal, bioplastics, healthy soil management, medicine residuals removal. With having that in mind, it is my great pleasure to introduce today's keynote speaker, Dr. Pete Myers. Dr. Myers is the founder and chief scientist of Environmental Health Sciences Foundation and an adjunct professor of chemistry at Carnegie Mellon University. He's one of the leading scientists on endocrine disruption, how certain chemicals interfere with our hormone system and affect our health. And this is a field that he has helped establish since the 90s. So perhaps for Dr. Myers, the novel entities are not that novel, as he's worked on them for 30 years. In 1996 already, he co-authored the book, Our Stolen Future. You will all receive a copy. It's a really well-written book, reads like a detective story. And that was the first book to bring this topic to a global audience. And it was a bestseller. More recently, perhaps you've seen the Netflix documentary, The Plastic Detox, where Dr. Myers was also featured. So we are certainly more aware of these issues, but it's also very clear that there's a lot more to learn, and more importantly, a lot more to do. So we are very excited to have Dr. Myers here today, all the way from the United States, to share his insights. Please join me in giving a warm welcome to Dr. Pete Myers.
Thank you. What a wonderful audience. Thank you for the greeting. I have a footnote to begin with. 1 year ago yesterday, I was supposed to be here, the year's last event. Instead, I spent my day on the operating table getting open heart surgery. I didn't plan that. And it led to almost a full year of recovery. And here I am. I think I'm recovered. Thank you. It's such a pleasure to learn about Wetsus. What an amazing institution. If I were a graduate student now, with both the dimensions of the science you do and the practical nature of the inventions that come out of this place, I would try and be here. Maybe I should try anyway. I've learned a lot about Wetsus speaking with all of you, or some of you at least. It is an amazing institution, and I congratulate the founders and all the participants in reaching so high and so far for doing the right thing with our planet. So thank you very much. I am going to talk about endocrine disruption. I should say that I'm on the faculty at Carnegie Mellon in the chemistry department, but I'm not a chemist. I'm a biologist. I began my work, my Ph.D. was at a marine laboratory in California, working on birds. And I won't tell the whole story about that transition. I'll tell a little bit of it. It has led me to understand the value of interdisciplinary research deeply. And to solve today's problems, I agree with Wetsus. You can't live in one discipline. You must cross disciplines. I'm on the chemistry faculty. I'm not one of those, in America we call them chemophobes, that is afraid of every chemical. We can't live without chemistry. Just stupid, silly to imagine that you can live without chemistry. But we've got to do it better. We have to develop a new approach to sustainable chemistry, and that's where my discussions will lead today. And we're working with my assistant here, Thank you very much. He's going to help me keep track of everything. Sustainable chemistry. What we need to be able to do is define what is safe and what is not. Most chemicals, most of the 300,000 or so that are in use in commerce somewhere on the planet have never been tested. Don't fool yourselves, they've never been tested, including most of the chemicals in the marketplace, in your supermarkets today, have never been tested. We have to figure out how to remove and destroy the bad things, like PFAS. And finally, we've got to design safer chemicals. And that's my role at Carnegie Mellon. It's not designing the chemicals myself. There are other people who can do that much better than I. But I can help them understand whether their products are going to be safe or not. You need to... Anyway, sorry. We can do this. I did write Our Stolen Future with Theo, Colborn, and Dianne Dumanoski in 1996. And we asked a question then. This is the subtitle of the book. Are we threatening our health and security and survival and intelligence with the chemicals that interfere with hormones? And the answer wasn't certain back then, but now it is. Yes, we are threatening our health, our sustainable future, and especially the future of our children and grandchildren beyond with the chemicals that interfere with hormones. This is Theo Colborn, whom I met in 1988. And she discovered in her work, she got a Ph.D. at the age of 53. Astounding woman. She knew how to connect the dots, to connect different disciplines, unlike any person I've ever known before or since. And she began her life as is this loud enough? Yeah. She began her life as a pharmacist with her husband, running pharmacies in New Jersey. And as she and her husband were doing this job, they realized they were killing their patients with the pharmaceuticals that they were delivering. They had not been tested sufficiently. So she went to Colorado and started a sheep farm, pharmacy, sheep farm. Interesting transition. And then, as she was doing that, she volunteered regularly for local nonprofit organizations that were working with water quality problems on the western slope of Colorado. And in that process, she went to a place called the Rocky Mountain Biological Laboratory. I don't know how many of you would have heard of that. It was an amazing place where leading ecologists of the country would go and spend the summer. Among them was Paul Ehrlich. He went there every summer, owned a house there. And Theo told me vividly about one afternoon she was there when she heard Paul, who became a good friend of mine, when she heard Paul and a guy named John Holdren, who was Obama's science advisor ultimately for both administrations, she heard them, as she called it, bullshitting, a tradition in the United States. And she realized she knew more than they did about the particular subject they were bullshitting about. So she decided to get a PhD. As I said, she got her PhD, she finished it, at the age of 58. An amazing bundle of energy. She did this work at the University of Wisconsin-Madison, and then started on a project looking at the Great Lakes, looking at water quality in the Great Lakes, running a project that her boss wanted her to identify cancer-causing chemicals in the Great Lakes. This was in the early 90s, and cancer-causing was a big bad word then, still is now. But she did that work, and she discovered that the problem in most of the wildlife, she was looking at the wildlife effects, most of the wildlife were not causing cancer. They were causing these strange defects, mostly that emerged at birth, like this cormorant with a twisted beak, fish with edemas that grew on their bodies just as after they were hatched. A whole series of problems, and she realized it was something having to do with their hormones. Now, I don't know how many of you have taken biology recently. You know, if you have, you know that hormones are about more than just puberty, more than just testosterone and estrogen flashes. Hormones are responsible for a whole lot more. They are really the symphony of life. What do they do? Well, obviously they turn genes on and off, but they also alter cell membrane permeability, so they affect the charges that different cells have. They activate and deactivate proteins. They control various physical processes, and they're responsible for metabolic effects like obesity. And finally, they regulate the circulatory system. So let me go into depth in one of these jobs of genes, turning genes on and off. As you know, gene expression begins actually before conception. It helps the fetus live through many, many stages of development. It helps the adult all the way through to aging and death. Hormones work by getting into the circulatory system, beginning with the glands where they're produced, and going to receptors somewhere in the body. If the cell it reaches has no receptors, it doesn't have an effect. But if it has a receptor for that hormone, it will turn on either cell surface membrane receptors or nuclear receptors. Some of you may have followed the Artemis launch these last 20 days. Imagine the numbers of contractors that are involved in producing the system that flew that rocket to the moon and back. Actually, my brother was part of that process. The hormones are akin to the signals that the contractors send to the people who build the rockets. You know, imagine what it's like to be a baby just born, and the numbers of, can you all see that? I can't tell what you can see, but the numbers of contractors that it takes to build a decent brain and a decent body, it's way more complicated than building the Artemis rocket. So hormones aren't poisons. They hijack a system that doesn't know the difference between the hormones naturally, and the system and the endocrine disruptors that substitute for them. It's a big difference in how any toxicologist looks at endocrine disruption. Endocrine disruptors can work in multiple ways. They can send the signal at the wrong time. They can send the wrong signal at the right time, where they can block the receptor process. And Theo built a solid database of studies that were building a story around the effects of endocrine disruptors. And she gathered a group of 21 scientists at a place called the Wings Break Conference Center in Racine, Wisconsin, in late July of 1991, based on all the work that she had done accumulating things that looked like they might be involved in endocrine disruption. And Theo was, I'm sure you all know a few people who connect dots where no one else has seen how the dots are connected. Theo worked that way. She connected dots. I was part of the organizing process for the Wings Break Conference Center. And it resulted in a text that became the foundation document for endocrine disruption. I actually made the mistake, or had the good fortune to coin the term endocrine disruption, because I had been working on climate change and realized that climate change was more than just global warming. It was all sorts of processes being interfered with. And I realized, as I listened to all these endocrinologists and other disciplines talk about the consequences they were seeing of the hormones that interfered with their studies, I realized that this phenomenon that Theo had pulled together was akin to global warming disruption, in that it had effects on many, many different dimensions of the weather system. And so I proposed that endocrine disruption become the operative term. And even though Theo objected at first, it stuck. Here she is with me and Dianne Dumanoski. Theo is the woman on your right. Dianne in the center. And we decided that by the mid-90s, we had enough information based on the many studies that were being published that somehow were related by the phenomenon of endocrine disruption. We decided it was time to take it public. We wanted to write a story that would then lead to public interest in the study and possibly funding. And so we wrote Our Stolen Future. We published it in 1996. And interestingly, I had become friends with a member of Congress named John Porter, a Republican. I could talk with Republicans back then. John was a Republican from Northern Illinois, and he became interested in the book I was working on as we were working on it. And I went to dinner at his house multiple times. He became really interested, and he happened to be the head of health appropriations for the National Institute of Health at the time. That's a congressional responsibility with huge opportunities to affect what NIH was doing. And so John, who became a good friend, invited me to go to the hearing process in Congress that he was chairing. And he also invited Harold Varmus, a Nobel Prize winning scientist who was the head of NIH at the time. He invited Harold Varmus to testify. This is what NIH needs now. Okay, typical congressional process. And right in the middle, John had me sit behind him. And right in the middle of the testimony, he leaned over and picked up Our Stolen Future. And he said, Dr. Varmus, you need to read this book. Now, Congress doesn't work by, at least it didn't used to work by, telling NIH what to do specifically. But when your chairman of your committee, your appropriations committee, says, be interested in this, learn about it, it sends a strong signal to NIH and science involved. Yeah, I better read that book. So it unleashed literally hundreds of millions of dollars in research money that went to NIH. And it was that bolus of funding that then enabled huge amounts of research to be undertaken by NIH, related in one way or another to endocrine disruption. And now, 35 years after the Wings Break Conference and 30 years after Our Stolen Future, we have a good idea about what endocrine disruption does. It interferes with all sorts of physiological and metabolic processes. The science on these medical conditions, none of it's certain. Science is, as you all know, how uncertain science remains and how close it is to get, how hard it is to get to certainty. But there are strong indications in the literature for each one of those conditions that support the hypotheses that endocrine disruption is driving them, or at least driving part of them. They also have genetic bases, of course. I bet the members of the audience here are looking at this word cloud and realizing members of their immediate family, members of their close collection of friends, members of their acquaintances, all are experiencing these various problems. These are today's epidemics of non-communicable diseases. They're serious. And endocrine disruption is not the sole cause, but is unquestionably contributing to elements of it. So I've got 5 key points that I'd like you to bear in mind as I continue talking. One is that endocrine disruption is not toxicity as it's usually thought. It's really hijacking hormone behavior. And what that means is that the usual defense systems that we have for overt toxicity don't work. They're irrelevant to this question. When you get endocrine disrupting compounds like PFAS at high doses, that's when toxicity sets in. But the effects of hormone disruptors, endocrine disruptors, takes place at much lower doses. Exposure is ubiquitous. I'm sure you saw the data that came out from a Swiss lab last year or the year before, showing that rainwater everywhere in the world has PFAS in it. PFAS is an endocrine disruptor. It's one of the multiple mechanisms that it works on. This upsets the toxicology community a lot. I'll go into it in a little more detail shortly, but high-dose testing misses completely the effects that take place at low doses of hijacking the hormone system. Misses it completely. So if you are testing at high doses and you find no effect, you may be missing the real effect of the hormone hijacking that's taking place. EDC, endocrine disrupting compounds, EDCs, do not have a threshold. They are operating on a hormonal system that's already functional, already working, already above threshold. And so you can add EDCs to it and you will see an effect. And friends of mine have worked out the Michaelis-Mencken equations that prove that to be true. Have any of you heard of transgenerational epigenetic inheritance? what it means is that you can inherit a disease without changing DNA sequence by changing the epigenetic control of the expression of that gene. And now we know that epigenetic inheritance goes down at least 4 generations of effects. I'll mention that a little bit later. So exposure is ubiquitous. EDCs are everywhere. They're in plastics, they're in soil, they're in water, they're in wastewater, they're in the air. You don't experience them all the same way because we have different exposures. But we do have exposures, all of us. I said this before, most chemicals, of the 300,000 or so chemicals that are acknowledged to be in use in chemistry today, commercial use, not just invented in the lab, those, only a small percentage of them, at most a few percentage points, have been measured for EDC characteristics. A lot of that is because traditional toxicological testing tests at high doses, doesn't test at low doses. The details of your exposures vary immensely in terms of the chemicals that are engaged, in terms of the amounts, and also the timing, and all those make a difference. Some of the exposures to those chemicals are essential. We need them for commerce as we have designed it today. We need them. I won't deny that fact. But I do challenge us to be thinking about, if we have a chemical that is an endocrine disrupting chemical and it's essential, can we design a replacement that's safe? And the answer is maybe. I mentioned that high-dose testing does not, misses low-dose effects. And let me show you a graph that a colleague of mine published a few years ago. Any of you heard of tamoxifen? It's a pharmaceutical used to treat breast cancer. It's an endocrine disrupting pharmaceutical that treats breast cancer. And here's what happens. I don't know how well you can see that graph in the background. Okay, but a typical toxicological test of something like tamoxifen begins at high doses and works down the dose response curve until there's no difference between the control, which is the line across, and the exposure effect. Okay, so what the EPA does, it establishes a no observed adverse effect level, the NOEL, and it says, okay, now we know where it doesn't cause an effect. And we're going to add some safety factors into the testing process. We consider that 1,000-fold beneath the NOEL is safe because there's a factor of 10 that's introduced to take into account that parents are different from their kids, that take into account their animals are different than the humans, and that there's variation in people anyway, and you call that number 1,000-fold beneath the NOEL, you call that safe. That's how all toxicological testing is taken. There are variations in it between Europe and the US, but that's basically the system. Find a level that's safe, extrapolate with some safety factors, and conclude that level, at the low level, has no longer any effect. The problem is if you extend the dose response curve. These are data produced by a scientist colleague of mine working with endocrine disruption that shows that at low levels, it stimulates the growth of breast tumors. After having found that it was safe at a NOEL, it turns out that literally at the point where it is most stimulatory for breast tumor growth is that 1,000-fold difference created by the safety factors. Actually, physicians know about this. If you have a family friend who's had to take tamoxifen, you'll probably have heard about the tamoxifen flare. That's the tamoxifen flare, where the tumor grows if it's maintained at that dose level. Physicians adjust their dosing to take that into account. They take people beyond that flare level as quickly as possible because it hurts. And so if you then take these data and ask, well, where is the quote-unquote true NOEL, no observed adverse effect level? It's actually 1,000-fold beneath where the flare is. And if you using the FDA's or the EPA standards, if you then calculate what's safe, it's vanishingly close to zero. This is why tests for endocrine disruptors have to look at across the dose response range. You cannot look at high doses only, even though it's really tempting, because you think that, oh, the dose makes the poison. Therefore, if I test at high doses, I'll certainly see the consequences of this exposure. But what you can see here is you miss the important consequence of this exposure, which is growth in the breast tumor. This is actually what you learn about in Endocrinology 101. Endocrinology knows there are some things called biphasic dose responses. It's basic endocrinology. Things happen at high doses that are different than those that happen at low doses. And I can go into the receptor biology, if you want, that explains why that's the case. It's expected endocrinology. Now, probably the most widely known example of endocrine disruption these days has been developed by Shauna Swan, whose photograph is there, and it has to do with sperm count decline around the world. And Shauna, I first knew Shauna in the early mid-1990s. She was asked by the National Academy of Sciences in the US to reanalyze Niels Kakebox. Niels is an endocrinologist in Denmark, and he had discovered indications of a long-term decline in sperm count, roughly 50% over 5 decades. And the National Academy asked Shauna to replicate that study, gather new data, and test. And Shauna, being an epidemiologist and very skeptical, said, sure, I'll do it. I'll finish the analysis in 6 months, and I'll be done. I'll go back to my job. She's still working on it, because after her first attempt to reanalyze it, she gathered new data, she combined them with the old data. She found that the slope of the curve did not change, even a percentage point. Niels Kakebox results were solid. She then repeated that study in 2021 with an Israeli colleague and found pretty much the same thing, except the decline was accelerating. And if you extrapolate the curve, of course, scientists don't like to extrapolate curves, this is a serious outcome. This indicates that sometime in the future, and not too distant future, reproducing the old-fashioned way ain't gonna work for many people. Greg talked about the Netflix documentary, Plastic Detox. That was Shauna's project that she designed. She said to herself, okay, I gotta do something. So let me do a pilot study where I provide guidance to a number of couples that have been proven to be infertile, medically infertile. And I'll see if I can change their fertility. And she did that, and I will give you I'll tell the end of the story. of the 6 couples that she looked at, 3 had babies by the time they followed her, after they followed her instructions. Now, that's a pilot study. We can't count on it as scientists. She's counting on it to get her a big NIH grant. But it's a pretty amazing result out of 6 couples, 3 babies.
What was the most important advice?
Excuse me?
What was your most important
Get rid of plastics. She went to each house multiple times and helped them cleanse their house. The question was, what was the most important advice? Thank you for asking that. And she went to each house repeatedly and did an inventory of what were the types of products they used, what did they eat, what were the ingredients of the things they eat, and she told them, if you stop using this, that'll help. And she tracked the sperm count in the men, and the sperm count in the men went up over those 3 months that she did the study, did the work. And of the 6 that she began with, 5 stuck through to the end, and there were 3 babies as a result of it. So who knows if this was a representative sample? They began as an infertile couple, okay? And they had 3 babies. Sounds promising. I know of one other couple that has done similar things after being medically infertile, and they did their own interventions, and they also produced a baby. Who knows? I think that the results of Shauna's new NIH work will help resolve that. So I think I may have convinced at least some of you that EDCs are effective at very low doses. Here's a simple graph showing you the dose range at which hormones work. And that's the yellow line. I don't know if the color's coming through here, but the yellow line is from low parts per trillion to medium parts per billion. That's the range at which hormones are effective at sending signals. If those signals of those hormones go above those ranges, there are protective mechanisms that set in motion that suppress the receptors. So that's part of the way that the receptor biology of hormones is non-monotonic, biphasic. The toxic range is that green dose, but EDCs are working down in the dosage range at which hormones are effective, and that's a hugely important point. Low doses matter a lot. Again, I'm emphasizing that. I don't know how much, can you all see this? This light is not good for me. These are 2 xenopus mating, 2 males. This is Tyrone Hayes' work. Some of you may have heard of Tyrone. This work was published in Nature. This is produced by 2.5 parts per billion in the water in which the males are growing as tadpoles. 2.5 parts per billion. And they wind up, the males are completely feminized, not just behaving like females, but producing eggs that can be fertilized. Okay, that's a shocker. The eggs that genetic males produce after being treated by 2.5 parts per billion of atrazine become functional females. And Tyrone Hayes, who's actually a friend of mine, and I was talking to him yesterday just so I got my facts right. He has a colony of xenopus, these are xenopus frogs, that is 6 generations full of male-only frogs, of which 10 to 20% have been feminized by atrazine and turned into functional females. Nature published this, okay? This is not made up. And if you just think about the dosage level, atrazine is applied, you guys are lucky. Atrazine is not used in Europe. It's produced in Switzerland, but it's not used in Europe. There's something to that story, but anyway, it's one of the most widely used herbicides in the United States. And it's applied in fields at about 1 million parts per billion. That's what the farmer wants to get. At about 8,000 parts per billion, you can find in running off of those fields. 80 parts per billion, you find in the surface waters of lakes that are downstream from where that runoff is occurring. You find about 1 part per billion in the clouds and rainwater that can be 50 or 100 miles south of the application site. Tyrone did extensive studies showing that. Those experiments were at 1 part per billion. The EPA in the US considers 100 parts per billion to be safe short term, and 3 parts per billion to be safe long term. And that's because they use old-fashioned toxicology to figure out what endocrine disruption can do to living organisms by hijacking the hormones they depend upon to, in this case, become reproductive. How am I doing for time? 10 minutes, okay. This is another study that's going to make at least some men squirm. These are the insides of mice, adult mice. They were treated with 20 parts per billion bisphenol A, another infamous endocrine disrupting compound. And this is actually the control animal. It's got kidneys that are the normal size. It's got bladders the normal size. They can pee normally. This is the result of treatment in utero when this exposed mouse was an adult. Its kidneys are blown, dysfunctional. Its bladder essentially will explode if the animal hadn't been sacrificed. And that happens because if you think of the urethra, which is the tube we pee through, it has it passes through the prostate gland, and the prostate has some tissues that are particularly sensitive to estrogen stimulation. And they set up a epigenetic pattern where as the mouse ages, the ratio of testosterone and estrogen changes. There's much less estrogen in an adult mouse. And the tissue around the urethra where it passes through the prostate gland closes like that around it, and the mouse can no longer pee, and so its bladder explodes. The point I would make here is that what happens in the womb may take a long time to develop in the lifetime of the individual. And this is the only medical model of obstructive bladder syndrome in people. We don't know for certain because they haven't been able to do the test yet, but they're working on it. This is work by Fred von Saal, another famous name in endocrine disruption. This may be the cause of that problem in urination. And I said earlier, I asked if you'd known what transgenerational epigenetic inheritance was. Fundamentally, here's what it is. Grandma gets exposed. Mom, the second woman on your right, is exposed in the womb if grandma were pregnant when the exposure took place. Granddaughter is exposed in the womb also, because she's growing eggs when she's still a fetus that become the reservoir of eggs that lead to her child. So you've got 3 generations of exposed people directly, and one that's not. There are now many studies of transgenerational epigenetic inheritance in animals showing that this can take place without changes in DNA sequence. It's almost, who's the, Lamarckian, it sounds, when you first hear about it. Remember Lamarck, the geneticist. But this actually happens. The reasons for it's happening are becoming more and more clear. And endocrine disrupting chemicals do not always cause it, but do cause it under some circumstances. And there are now enough studies in people showing that this transgenerational epigenetic inheritance clearly goes through the 3rd generation. And right now, studies are underway to test the 1st generation that was completely, at least unexposed, according to measurements of umbilical cord. The last example here was very recently published, and it has to do with autism. And it has to do with low doses of bisphenol A causing changes in the behavior of aromatase genes. It suppresses them, and it leads to an increase in testosterone. Testosterone is a product of that suppression of the genes. And the brilliance of this study, which was published in 2024, was that they not only did the mouse work with both exposed animals and knockout animals for the aromatase gene, but they also studied about 1,000 kids and looked at bisphenol A levels in kids and discovered that at 2 years of old, the baby boys, this did not work with females, the baby boys were a factor of 2 to 3 times more likely to have autistic characteristics. But at the age of 9, they were 6-fold times more likely to have autistic characteristics. And highly statistically significant confirmation of the effect of aromatase suppression in the mammalian brain development. So, just to have a little bit of fun, are low doses really real? What's a part per billion in terms of numbers of pancakes? 1 part per billion is about 1 pancake in a stack of pancakes 4,000 miles high. Sounds like a little amount. But if you look at that in a different way, you look at a drop of water, and you calculate how many atoms of BPA are in that drop of water if it's at the concentration of 1 part per billion. 2.65 trillion molecules of BPA are in that 1 drop of water. 1 billion times more molecules are in the drop of water, or in that drop as water, but it's still a lot of molecules if these molecules are capable of acting at very low levels. So I'm going to close with a few comments on what would sustainable chemistry look like. And I think the first question we have to answer is, what is safe and what is not? And then we can get to the questions of, well, if it's not safe, how do we destroy it? And also, how do we design safer chemicals that can help us avoid these problems in the future and take care of future generations and protect them from exposures? This is a chemical company. I'm on the board of a chemical company, believe it or not. I'm on the board of a chemical company that was whose technology was invented by a colleague of mine. This is Terry Collins, he's the inventor. He started thinking about making peroxidase enzymes artificially that could be used commercially in 1980. He started working with that in 1980, designed a process by which he could tweak molecular boxes that were imitating peroxidase enzymes, make them smaller and smaller and more effective. And the iterative process that he worked through led to catalysts that now are thousands of times more effective than the native peroxidase enzymes, and small enough to be commercially producible. And that's the product that Sudoc has brought to market. The processes that, for the most part, have been used to oxidize bad chemicals are expensive. They use a lot of water. They use a lot of energy for providing the heat. That means higher costs, a bad impact on the planet, and also dangerous work for the people who have to use them. Terry invented something called TAML and new TAML catalysts and put them together in a company that's called Sudoc, or Sustainable Ultra-Dilute Oxidative Catalysis. I can only say it slowly as a non-chemist. But this oxidizing set of catalysts work pretty amazingly. They work with they catalyze common oxidants like hydrogen peroxide and hypochlorite. They dramatically increase the efficiency of the oxidation process. They can be added to existing commercial products. Without having to induce, to build big infrastructure changes in the process. And they enable a lot of water saving, a lot of energy saving, and safe conditions for people to work with. If you want to learn more about this, invite Terry Collins, who's a brilliant lecturer. He is a real chemist. He worked on that, inventing that process, like I said, beginning in 1980. He had the first TAML in first functional TAML in 1995, and then he introduced the second version of TAML in 2020 and invited me to join him in commercializing the company. Oh, by the way, I gave all of my shares to that company to a nonprofit, so I don't benefit from that at all. I think the dynamic between chemists making money and not being willing to step away from dangerous products because of that has been one of the big drivers of the chemical world we live in today. And I'm exceptionally proud of the fact I don't benefit from the success of this company. So we have a new day in front of us, I hope. We can actually reduce the use of bad stuff. That's a technical term, bad stuff. We can redesign the chemicals using the types of approaches that Terry Collins used to develop TAML. And we also can work with the regulators if they will begin to think seriously about the new science that's available on endocrine disruption and other compounds like that. So thank you very much.
Thank you very much, Pete, for your very interesting keynote. You painted a very worrying picture, but thank heavens you gave us some hope in the end. And we've grown quite accustomed to getting bad news from the United States in the past year and a half. But this was
not my fault.
This was a very different level, and thank you for it not being your fault as well. So I can imagine there are some questions about this intriguing story. Professor Buisman has the first question.
Yeah, I would really like that you give my young colleagues a tip how to increase their sperm quality.
Watch Plastic Detox on Netflix. But there are some simple things. I wrote in 1996 in Aristotle in the Future, don't microwave in plastic, period. Doesn't matter whether it tells you it's safe for microwave or not. Do not, in 1996 I wrote that. And now you'll read it everywhere, and it's true. The heat increases the rate at which bad chemicals leach out of plastics, and there are 16,000 chemicals that are known to be used in the manufacture of plastic around the world. Very few of them have been tested at all. Don't microwave in plastic.
So a new measure here at Wetsus,